Hello and welcome back to Teach Me in 10. In this episode on mitochondrial toxicity in the drug discovery process, we're joined by Julie Eakins, Principal Scientist at Cyprotex, who's here to walk us through today's topic. Over to you, Julie.
Hello, I'm Julie Eakins. I'm a Principal Scientist in the Toxicology Department at Cyprotex UK. I've been here for 11 years. Prior to that, I was an in vitro toxicologist for a big pharmaceutical company in the UK. Today's presentation is going to be around mitochondrial toxicity, the implications of such toxicity during the drug discovery process, and I will show you some of the assays that are available for early screening to help de-risk this phenomena. So today, I'm going to demonstrate the integrating mitochondrial toxicity screening into early drug discovery. So as you may know, mitochondria are very important organelles. Generation of energy via a process called OXPHOS is their most important role, since they generate over 95% of the cellular energy in animal cells. However, they also host a variety of other key metabolic functions that determine cell function and fate. Our knowledge of mitochondria has expanded in the last decades, and it's now clear that they participate in nearly all aspects of cell function, affecting processes including cancer, inflammation, metabolic signaling, and cell death. Given the key role that mitochondria play in metabolism and cell survival, they represent an attractive target for many antibiotics, fungicides, and for many poorly treated common diseases such as metabolic disorders, neurodegenerative disorders such as Parkinson's and Alzheimer's, cardiovascular disease, and cancer. Mitochondrial toxicity has been considered an important mechanism of toxicity for a number of years and has been implicated in high-profile withdrawals from the market of a number of drugs, along with a number of black box warnings with huge financial implications. In particular, it's been implicated in multiple organ toxicities, such as hepatotoxicity, cardiotoxicity, and nephrotoxicity, as well as a potential contributor to idiosyncratic drug-induced liver injury. This table shows several clinically used drugs with black box warnings from the FDA for hepatic, cardiovascular, and renal toxicity. Those with mitochondrial liabilities are highlighted in red. Perhaps two classic examples of this are the anti-diabetic drug triglycerin and the lipid-lowering agent cerevastatin. Both drugs were withdrawn from the market due to high rates of hepatotoxicity and rhabdomyolysis, respectively, which was later linked to adverse effects on the mitochondria. As I mentioned before, mitochondria are often referred to as the powerhouse of the cell, as approximately 95% of ATP production in animal cells occurs via a process called OXPHOS. In this process, nutrients such as glucose, proteins, and fatty acids are metabolized into electron-rich substrates that act as electron donors to complexes located in the mitochondrial membrane, known as the electron transport chain, or the ETC. In the ETC, the transfer of electrons from substrates to oxygen is coupled to the translocation of protons from the matrix to the intermembrane space, generating an electrochemical potential gradient of protons, which is used by the ATP synthase to generate ATP. Hence, oxidative and phosphorylation are linked. A major focus of our in vitro approaches has been the disruption of OXPHOS by chemicals, which is a very common mechanism of mitochondrial toxicity. Disruption of OXPHOS can be caused by direct inhibitors of the ETC or by uncouplers, i.e. chemicals that cause dissipation of the proton motive force generated by the respiratory chain. Here we show some of the possible in vitro approaches which can be taken to assess effects of mitochondrial function. Further elaboration will be given on the items that have been highlighted, and I will go into these in more detail now. One of the earliest screening assays developed was for the glucose-galactose assay, also known as the GLUGAL assay. This assay uses ETG2 cells, an immortalized liver-derived cell line that primarily relies on glycolysis for energy production, despite having functional mitochondria. The assay exploits a phenomenon known as the Crabtree effect, where high glucose concentrations suppress mitochondrial respiration. By switching the culture medium from glucose to galactose, the cells are forced to depend on oxidative phosphorylation, thereby increasing their reliance on mitochondrial function. The assay compares compound toxicity under high glucose and galactose conditions. An increase in toxicity under galactose suggests the presence of a mitochondrial toxicant. The graph on the right shows an example where we show entacapone has demonstrated a tenfold shift in toxicity under galactose conditions compared to glucose conditions, indicative of a mitochondrial toxicant. We can build on these simple assays with the more complex and specific assays which are now available that allow the direct measurement of mitochondrial function. One such platform is the Agilent XFE96 flux analyzer, formerly known as the Seahorse, which enables real-time assessment of mitochondrial respiration and glycolysis. This system also allows us to investigate the effects of mitochondrial complexes, helping to elucidate potential mechanisms of toxicity. I will describe these assays in a little more detail shortly. Additionally, we can utilize high-content imaging techniques to evaluate the impact of compounds on mitochondrial replication. This is particularly relevant for compounds targeting viral or bacterial replication, where mitochondrial interference may be a potential concern. So the Seahorse assay, or the mitochondrial stress test assay, enables the assessment of mitochondrial function by measuring key bioenergetic parameters in real time. This is achieved through the sequential injection of mitochondrial stressors, oligomycin, FCCP, and a combination of antimycin and rotenome, onto the cells using a specialized cartridge. The cartridge contains 96 sensors, one for each well, with both oxygen and pH probes, allowing the simultaneous measurement of oxygen consumption rate, OCAR, and extracellular acidification rate, ECAR. Initially, the compound of interest is added directly to the cells to assess the impact on both respiration and glycolysis, providing a baseline measurement of cellular metabolism. The subsequent injection of the stressors allows for the dissection of specific mitochondrial functions and allows us to determine ATP production, proton leak, spare respiratory capacity or reserve capacity, maximal respiration, and non-mitochondrial respiration. The graph shows a representation of the stress test, which is performed by sequentially injecting oligomycin, FCCP, and rotenone antimycin through the injection ports. These changes in profile can be used to identify potential mechanisms. So knowledge of compounds capable of poisoning mitochondria, such as the classic ETC inhibitors, goes back more than 60 years. And today, thanks to OCAR, ECAR, and reserve capacity measurements, we can identify mechanisms by which unknown compounds induce mitochondrial impairment. And this slide shows a few examples. In the top left is an example of a classical ETC inhibitor, where we get a decrease in oxygen consumption rate, a decrease in reserve capacity. And due to the block on the OXPHOS, we have an increase in extracellular acidification or glycolysis to compensate. In the top left is an example of an ATP synthase inhibitor, where again, we see a decrease in oxygen consumption rate as expected, but this time we have no effect on the reserve capacity. Again, though, we do see an increase in ECAR as a compensatory mechanism. In the bottom left is an example of an uncoupler, such as 2,4-dinitrophenol, which caused the dissipation of the protomotive force and stimulates substrate oxidation and inducing excessive oxygen consumption. So we see an increase in OCAR. But as this is not linked to oxidative phosphorylation, we also still see an increase in ECAR. Compounds with other off-target rather than mitochondrial toxicity often shows a decrease in all three functions, OCAR, ECAR, and reserve capacity. Once we've identified a compound which inhibits oxygen consumption rate, we can follow this assay, as it is possible to identify which of the individual complexes are involved. In this assay, we still track real-time changes in OCAR, but since many oxidizable substrates, such as succinate, are unable to cross the plasma membrane freely, and additionally, many cell types can store and oxidize endogenous substrate, it's difficult to control and determine which metabolic routes are fueling respiration. So we use selective cell permeabilization, which offers solutions to this without having to isolate mitochondria. In this assay, we permeabilize the plasma membrane of the cells, and a combination of different metabolic substrates and inhibitors are used as an approach to study mitochondrial function in situ by exploiting the fact that oxidizable substrates feed differentially into the mitochondrial pathways. This slide shows how we've proven the assay to work using a number of specific well-known ETC inhibitors, and the graphs show the trends in the oxygen consumption rate in the different respiration states. So we start with complex 1 respiration. At the beginning of the assay, respiration is fueled through oxidation of complex 1 substrates. If the test compound decreases OCAR at this point, this indicates the chemical in question could be an inhibitor of either complex 1, 3, or 4. With complex 2 inhibitors, we don't usually see an inhibition of OCAR at this point because mitochondria tend to bypass complex 2. We then add succinate and rotenone to induce complex 2 linked respiration and inhibit complex 1 respiration. If at this point we see a recovery of OCAR to the vehicle, this would indicate the compound was a complex 1 inhibitor. If no recovery is seen, it suggests that the inhibition is either complex 2, 3, or 4. If rather a sudden drop in OCAR is observed, which was not seen in complex 1 respiration, this is a strong indication of a complex 2 inhibitor. Finally, we add ascorbate, TMPD, and antimycin to induce complex 4 linked respiration. At this point, we can then differentiate which compounds are complex 3 inhibitors if we see a recovery, and which ones are specific complex 4 inhibitors with no recovery as observed. So finally, we have demonstrated that mitochondrial toxicity is an issue associated with many target organs. Implementing an early screening strategy can reduce the risk of late-stage failure. Assays have been developed which are amenable to implementing in early drug discovery, such as the GLUE-GAL assay, the mitochondrial membrane potential assay. Assays have been developed to measure mitochondrial function directly, such as the seahorse assay, the permeabilized cell assay, and the mitochondrial biogenesis assay. Thank you very much for your time.
Thanks, Julie, and thank you for joining us for another episode of Teach Me in 10, which is brought to you by Cyprotex, an industry leader in vitro and in silico admitox. If you enjoyed this episode, be sure to like and subscribe to catch all of our latest releases.